Assalamu alaikum rahmatullahi wa barakatuh. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. Inshallah ta'ala, I'm briefly going to go through or bismillah ta'ala, try to go over the uh, BIOA02 Lab 1 to 3 review. So this will be considered week 7 material bismillah ta'ala for the next 2 hours. May Allah subhanahu wa ta'ala accept it from us all. Could someone, oh, sorry. Hi everyone. Good morning. Give me a few moments. Apologies. Really appreciate the patience. So I really appreciate your patience. Will you be recording the Zoom to look back to? Yes, Amina, great question. So as we speak, it's recording actually. So I hope to inshallah go over the material and for those that perhaps want to have a look back, they can. But I do want to stress the importance or hope that everyone will pay attention during that time. Uh, by the way, who is iPhone 180? Just so I can get context. Oh, Nia. Okay. So, mashallah, assalamu alaikum wa rahmatullahi wa barakatuh, both of you. So let's go over some of the material and just for context. Um, wa alaikum assalam wa rahmatullahi wa barakatuh. I know that probably you guys are, you know, perhaps enjoying Eid if you haven't, Eid Mubarak in advance. But I know like these past few nights or few days, inshallah ta'ala, you have some fun, inshallah. Alhamdulillah, it was good. It was good. Alhamdulillah, it's always good. I actually went to campus, so we had some Eid Salat. We had Eid Salat there, and we had some people attend. Alhamdulillah, it was really nice. Um, uh, and pretty much I walked in and out from home to campus, which was also fun. So, and a bit of reflections from there on. So, alhamdulillah, it was nice. How about for you guys? I know it's a bit of a tangent, but how was Eid Salah for you guys? Yeah, awesome. Alhamdulillah. And that's really good to hear. If once again you guys need any accommodations, I mean, it's a bit different for me in the sense that as a TA I would be able to understand. But if it's, let's say, for an upcoming term test or like at least in Ramadan, you'd reach out to the coordinator of your courses, and then inshallah they hopefully would accommodate accordingly. Um, and similarly, even for today, if you're, let's say, having your Eid today and you have a term test, you can request for there to be a deferral or extension. Um, I think I'm having, okay. Okay, so we'll talk about stats. Okay. Okay. No problem. No problem. Inshallah. Also, Nia, if there's any things that you want me to go, like, so let me tell you what my plans are. I was going to go through, I know we're a bit tight on time, but at least inshallah before one, I want to go over the content for weeks 1 to 3. And so inshallah, the goal is spend about half an hour or sorry, I think I said 50 minutes. 15 minutes, half an hour, about half an hour per topic. And then inshallah leave the rest for Q&A, either in between or at the very end of the seminar, inshallah. How does that sound, by the way? You can put like thumbs up, emojis, or share your thoughts accordingly. Yeah, perfect. Okay. And let me see. Yeah, so I'm gonna try to go through the topics and then see like topics that I want you guys to start thinking about, especially in prep for the lab exam. As we are aware, there are different questions, different formats of questions that will be posed to you, right? And it's timed for an hour, if I'm not mistaken, with 9 to 10 questions. So try to think about, distributing your time well in terms of the different topics, concepts, analysis that you've done in the past, and what you could thereby essentially um, interpret and make use of or apply and translate towards your final lab exam. So, any questions, by the way? Okay. May also just remove any weight rooms. I don't know if just in case someone comes by. I don't think anyone's here. So, apart from you guys, well done also for coming in. And so, if you have questions, let's actually go straight ahead to the content. So, okay, as you can see, there are different, oh, this photo is not showing, pretty much of eudicots versus monocots and their morphological arrangements. I'm just skipping through to show you, to focus and maybe give you a brief preview as to what we'll talk about in the next 20 to 30 minutes. And maybe just to bring your attention, if you want to look at the lab manual to see the exercises themselves, this will also help solidify maybe certain concepts that you may want to go over or questions in which you have for me now, inshallah. So let's go with, our first exercise with FLR. I'm also going to try to time myself just so that I can keep track. Um, okay, so when we look at, for example, exercise 4, we are obviously dealing with a plant, right? And plants have different, you know, um, metabolic, nutritional demands. And so, with these different concepts in mind, and probably what you've discussed in your FLR, and what I briefly want to highlight as well is that if anyone has outstanding questions um, regarding this assay, we could discuss them as well. But the importance is that essentially, you had a student set up for this assay or this experiment to essentially see the effects of drought on winter wheat. And bear in mind that, as I've talked about before, you have the common and scientific name. And so you want to just keep that in mind. أعوذ من الشيطان الرجيم. رب اشرح لي صدري ويسر لي واحلل عقدة من لساني يفقهوا قولي. Let me just go straight ahead now. Now that I've talked about a bit of, the bioassay, um, you have essentially um, different conditions, different treatments that we've analyzed, right? And we split out into groups from the entire lab, but you had reps, two reps, i.e., replicates for the control treatment, the drought treatment one, and drought treatment two. And thereafter, control treatment is assumed to have no drought and be watered regularly every day, i.e., and then drought treatment one was watered every other day. And then drought treatment two was watered every third day. I want to make a note that some people may have mistaken this in their FLR, thinking that drought treatment two with the number two would have been watered every second day, but technically every other day and every second day is the same. So the distinguishing factor is that it was watered every third day. So keep that in mind. And another separate point, but when it comes to treatments, even though technically we spoke about the effects of drought, on a controlled treatment. You want to think also, to what extent would you water these plants, right? Because yes, they're growing up, right? But there's also you needing to keep in mind, like as you maybe have plants at home, do you water them every day, right? Like technically, these are starting off from like, seedlings or even before then, like to be able to develop into seedlings. But you want to keep in mind, in those next 14 days, should you technically water every single day, or should there be slowly an interval between which you're watering, such that you're not overwatering them, right? So you could actually think about those effects. Um, yeah, tomato plants as well. Jazakumullahu Khairan. Great point, Almina. And so these are things that you want to consider. Like winter wheat, actually, you would likely do it almost every day, perhaps for the first week. But the second week, it's likely to change that schedule. So you want to consider that as well as a potential limitation. But then moving forward now, and I hope that makes sense. It's really the different. Before I even move further, I'm going to even move back just to highlight something. In your lab exam. Maybe I'll just open it up. The previous, just to go all the way down. Because I think it's very important to understand the relationships between biology and reality, right? So, or like theory and biology in the sense, or in biology, comparing theory with reality. And I say this because, like, as you could see, there is a chunk of material that refers to, demonstrating how you could interpret certain stats or values and the relationships between results that we've done thus far. But you also want to think, and even for your future labs and exams, the connections and interpretations, and more importantly, the applications that you're able to interpret for different topics. And so, even some life skills or some key skills I want you to take away, even from our lab together, is one, as I talked about before, integration, right? Implications, investigation, and innovation or inventions. And I incorporate this from an Islamic perspective because I find that at the forefront, all of this belongs to Islam, and really, like as a Muslim, portraying as such with the best of conduct, content, and confidence is something that I want to build, and inshallah, all of you will as well. So I just want to bring that up because this was also part of my reflections yesterday, post Eid. So I think that perhaps it'll be a benefit for everyone as well watching to feel like they can actually apply the material that they've learned and see how it could be a better use for humanity beyond biology. So, so inshallah, let's continue. And I want you to really think about those skills that I talked about. Um, I'm actually gonna write them down. So oh, actually I don't wanna have only 14 minutes. So let me go briefly. Let me go continue, inshallah. But setting up your exercise, maybe we'll talk about certain concepts in due time that I've touched upon, like those skills that begin with the letter I. And pretty much in an acronym is called the I-5 approach. You pretty much set up these containers, and something that I was keeping in mind is that, are you adding in the right volumes, right? In terms of your methods, so I'm connecting back to the FLR, right? There is like a sequence, schematic shown here, right, with the different layers that you could see on the slide. Um, yeah, Jazakumullahu Khairan Alameen. I really appreciate continuous points that you're pointing in the chat. May Allah bless you. What I'll say is as well, like connecting back, no problem, no problem. Um, when it comes to essentially looking back at the methods, you want to consider, oh, it just slipped my mind. So let's actually go ahead for timing. I'm gonna pause the timer, make an edit. Okay, so now we should have a bit more time. The point of really this essay is bearing in mind the sequence, the order, and the right amount of, and essentially following suit with the methods that was described in the manual. So that way, especially when it comes to questions relating to specific concepts or controls, then you should be able to understand where the question is coming from or how you could best answer those questions. For example, let's say you were given a situation, and I'm not writing the exam, but I'm just giving you an example hypothetically. Let's say your container was filled with was done in this way, but there was a third layer of paper towels on top of the fifth. So actually, technically, four layers. Um, that would likely impede, right, the growth rate of your wheat seeds, or if and then thereby reduce the amount of shoot length that you would see on average across um, your control or your other experimental treatment groups. In contrast, let's say you added 100 mils of wheat seeds, right, hypothetically, or 20 mils. Then, of course, you would thereafter see a great change, right? Exactly. There is an additional barrier in place. And so those are things that you want to consider in mind, and even controlled conditions. Um, with humidity, temperature, light exposure. These are things that we've also connected with the Daphnia isopods, etc. But I hope, and even the Lamna experiments as well. So I hope you, inshallah, can appreciate this and connect back to what I talked about before and about Western blotting, because it's a very similar concept as well. But sorry, I'm going on tangent. So let me go straight ahead to exercise two. And if there's questions, I'll answer them ta'ala at the end. But you can make a note of them in the chat or in your notes. When it comes to now morphology, and by the way, our exercises are framed between like exercise two to the very second last exercise because the last exercise is the lab assignment and the first one is your lab quiz. If you have questions, we could discuss them, but I would primarily focus on the material shown here. Generally, as you could see from the manual as well, we have differences in morphology, in different patterns and components or pairs of components that you find within flowers. Angiosperms are flower-producing organisms, and then gymnosperms produce seeds, if I'm not mistaken. Um, Now, when it comes to your flowers, they are separated into four distinguishing categories or components known as whorls, and whorls go from down to up in number. So the top whorl, which is the pistil, the female's organ, is actually whorl number four, and then you have the other ones that descend thereafter in lower decreasing number. But simply you have the sepals, and this is something to keep in mind. If you're able to distinguish them, sepals are normally underneath, the petals, right? So and you can think of it in alphabetical order. You have petals before sepals, right? But in terms of where their arrangements are, the sepals are underneath as whorl one, and then whorl two is the petal, and then or the set of petals, and then you have the stamen as a male reproductive organ, and then the pistil as a female reproductive organ. And you could see and notice not just the differences in morphology, but in abundance. And this also connects to like the male reproductive and female reproductive systems of humans, right? Because generally speaking, there's a limited number of or quantity of eggs produced in the ovum. And then you have, in contrast, like many, like millions, if not billions, of sperms that are essentially present prior to fertilization. So there's indeed like integrations that you can connect the dots with, looking at, the flowers, like different angiosperm reproductive sets, and then even comparing the morphology with what we see in what do you call in ourselves, right? In other organisms, and that's hopefully something you'll connect the dots with from the zoo as well in terms of differences and patterns. But briefly, angiosperms, you have differences in propensity uh, of different, in at least this category, differentiating monocots from eudicots. Anyone know the difference? You can feel free to text it in the chat. You can notice though that 23% of them are monocots. Sorry, I'm speaking a bit too quick, so I want to let you guys write down the difference. Or you can also talk. That also works indeed. Okay. Yes, you know the difference. Monocots have actually no. Monocots have fewer. So let me explain. Monocots are well, in terms of propensity, 23%, and unicots are 75. But the difference is actually monocots usually come, and I know it's not shown here, but monocots, generally speaking, come in sets of three. So what that means is that their sepals and their petals, or their petals, you could consider them separately. Their petals could be in um, like essentially a total of three, or a total of six, or a total of nine. Whereas for eudicots, they come as sets of four to five, meaning that eudicots, and this is something to help distinguish them, will normally come in sets of like four, eight, or 12, or if you're going by pairs of five, then it could be 10, 15, or five, right? So you want to consider like the differences um, in their arrangements. Yeah, and mono also gives a hint as well. So you'll kind of appreciate these differences. No problem, no problem. Great response. Thanks for your response. I really appreciate it, Almina. So, but continuing now, hopefully that's something that you want to be able to distinguish. And then if you compare here, right, like the symmetry is also something to bear in mind. You have radial versus bilateral, in other terms, acidomorphic and zygomorphic in terms of symmetry, right? So this actually will likely be familiar if you recall from elementary school. You have like some letters that you use a mirror. I don't know if you've learned this in like elementary school, but during my time, you'd have a mirror, which is essentially a like glass, plexiglass looking mirror where you'd be able to see a reflection, let's say, of a letter. And so, like an O, for example, would have radial symmetry, generally speaking, or like a circle. A circle would exhibit radial symmetry, which is where you see that term come in. And then bilateral is where essentially you would have a letter or a plant in this case split up into even halves. So this is a good example, but like if you're looking at letters, the letter A is an example, the letter I is an example, um, and you have some others as well, right? Or letter H, and those are just an M, so some of them are actually reflective of my name, kaleidoscope. Um, Yes. Great response as well. See, the connections you'll probably take into account in many unique ways. And so those are things that you want to keep in mind. Um, Even in organic chemistry, there's a concept known as, and hopefully you're able to remember, um, you have stereoisotopes, or you have essentially, um, you have certain compounds that are also similar in being able to reflect on either side, right? So those are things that you will have also not only like biological relevance, but also chemical and beyond, right? Even from elementary school. So that's something to connect the dots with. And then once you're done that, well, actually, during that time, you'd be looking at the dissecting microscope, so the one that we saw here, right? And the idea is that you would have actually, as you recall, perhaps, you would have essentially identified at the whole level surface, right? So this is like essentially you looking at the outer surface, trying to see like here the different components. So you have, for example, the stamen, you have the pistils. Let me just also confirm with you, yes. So you have the pistil in the middle, and then you have the stamen with the pollen grains on the top edges. And this connects us with exercise 3 as well. But briefly, you would essentially, as we've done before, annotate your figures to draw them out from what you saw in the dissecting microscope or even at hand, and then inshallah annotate the different features that were shown here, and also classify based on the different symmetries or configurations or the number of sepals or petals, then to derive the essentially classification for eudicot or monocot. Let me ask, is this a eudicot or monocot, this plant? And what type of symmetry would it have? Just so I could get a feel. Maybe I'll say it then. Oh, so essentially, it would be you'd. I will let you know. It's okay. Um. Oh, let me see. Great job. You did caught. And in terms of symmetry, I'll let you know. It's radial. So think because generally speaking, even though it's four, if you cut in either like halfway in either way, like into either quarter, you should be able to see the other halves reflected, or the other quarters. Like if you split in half in any direction, you generally should see the other side similar to the one that you like on either side. They should both be relatively similar in shape. And that with that in mind, right, you want to have those different components listed, but also you want to highlight importantly in your figure caption, like outside of like beneath your figure, in this case a diagram, a detailed, right, annotation of your figure. And this is something that I want you to consider, right? You normally have like a title. So let's say this was my figure. I'm gonna say figure one, visualization of um, morphological traits or features in, so I'm going to give a specific name, Arabidopsis. Arabidopsis. This is a common uh, type of plant that is used. You may even have heard of it from maybe Dr. Von Estelic. Yes. And that's another point. Yes. So typically you refer to both the common name. So let's say this was a I forgot the name the type that it was, but let's just say it was um trying to remember. I forgot the type it was, but essentially it's a specific type of angiosperm. Let's just say it was a specific type of flowering plant. You would include the common name, and then you would say in brackets the scientific name, and then go over and using a dissecting microscope, right? Because you want to highlight those details as soon as you can, and then highlight other components, right? Like what you saw, how you prepared your sample. Actually, normally you would say how you prepared your sample first, your sample, etc. And then those details and any extra features that would be required of you. But that would really help to give you an understanding, hopefully an appreciation of the um, making a detailed figure caption, right? Because I'm not sure if you may have to consider that. But nonetheless, when we continue exercise two, you recall there was a second figure, and that's where you actually did the dissecting process. So you would have seen a cross section, pretty much splitting the specimen in half, and you would have hopefully been able to see a thin layer of ovules, or an ovule within which eggs were found. It may have been difficult to see the eggs themselves, but hopefully you would have seen the internal lining within the ovary, which would actually exhibit the ovule, i.e., the location of where you would find the eggs pre-fertilization. And then let's just see. Yeah, and then so you would have highlighted very similar components as before. Um, Now let's go to exercise three. I notice I have limited time, so I'm just gonna go to exercise three and highlight as a second component briefly in terms of not just transportation. I think what we've really focused on was on the diameters. So taking these details into account. And I know, yes, so let's take a moment back. We saw different angiosperms, right, from the figures before, and hopefully we could appreciate that as they have different morphologies, they have different abundances, even of their stamen, the male reproductive organs. And so something to keep in mind is that, okay, how does that impact even pollen migration? And in this regard, in a similar level, we're looking at pollen diameters because if you have larger pollen grains, then the idea is that they could impact not just the transportation, but the migration of pollen from one flower to another for fertilization or for growth and development. And so those are things that also connect with the previous slide because spore formation exists and you essentially have the formation of the male gametophyte. But moving forward though, sorry, I don't want to tangent back, but you actually have different categories that you're able to explore or different options of categories. So you could have explored the differences between monocots versus eudicots, angiosperms versus gymnosperms, insect versus wind-pollinated pollen grains, and those are things that you would have considered, right? Um, and um, and you would have seen within your lab manual, I believe. I could briefly show you the name of it. I think actually it's relatively simple if you wanted to look back in lab one. But the idea is that hopefully you were able to use a table of reference to denote, okay, the morphology or based on the color and the shape and size of your specific pollen grains, what would appear to be of essentially comparison. And then you would have had your research hypothesis that would have narrowed down on this concept. Um, and then briefly now, you would have calculated your pollen grain diameters, right? So, and normally I tell people to take the longer axis, but this is something you want to keep in mind for a lab exam, accounting for differences of subjectivity or accounting for differences where someone maybe took another axis for measuring, let's say, the pollen grains, maybe from their width rather than from their height, for example, or maybe the pollen grains were oriented in a unique way on your slide, right? So those are things that you want to keep in mind. And then inshallah, using the calibration factors already made here, you would essentially account for the size in OMUs. So this looks like 15. 15 at, let's say, a 10 times magnification, which is something you want to keep in mind, by the way. So you're at low 10 times. So you need to know the mag to be able to determine the ratio. But because it's 15, you would multiply 15 OMUs by 25 in the calibration. It's divided by OMUs multiplied by micrometers or microns. And so 15 times 25 will give you 375 micrometers or microns. And so as you can imagine, that's very, very small. That's actually like almost 40% of a millimeter. So just imagine how small these pollen grains are. And even that's, by the way, a larger one, right? Perhaps. But nonetheless, you want to consider those effects because they may have differences, right? From like wind to insect pollination, because wind would travel generally smaller pollen grains, whereas insects can carry like ants, ones that are larger, even like double or triple or quadruple their size. So their own mass. So those are things that you want to keep in mind. Those could be like seeds, for example, which are generally more larger than pollen grains. Now, briefly, we have stats, and I'm going to talk about this briefly in the sense that you have, once you are able to, state your null and alternative hypotheses, you would hopefully be able to appreciate, okay, if there is no effect or no observable uh, difference or comparable difference or significant difference between pollen grains pollinated by, like essentially from monocots versus eudicots, or from insect pollination versus via insect pollination or wind pollination, etc. So you want to consider like if it's null, there's no like there isn't sorry. In the alternative, you essentially find that there is a comparable significant difference. That's actually where the stats come in, because after you've calculated the mean for each group, that would have had the same numbers, and then determine the variance, standard deviation, and 95% confidence intervals, which you have for formulas in your lab notebook, in your manual from the tables shown. You should have hopefully been able to thereafter prepare a graph with the means and 95% confidence intervals. So that way, if they actually overlap, indicating where essentially or how close the means or different categories are to one another. If they did overlap significantly, then you would fail to reject the null hypothesis. Sorry, you would, yes, you would essentially um, accept the like, or you would assume the null hypothesis, but if they don't overlap, then there is assumed a significant difference with a p-value less than 0.05, but generally speaking shown here as 95% intervals that don't interlap. So that would essentially highlight your results and your stats. So and then what I normally tell people is that you want to think thereafter, what are the interpretations, what are the implications of those results, right? So if seeds that are pollinated by eudicots or that are from eudicots are larger in size versus monocots because they may have, let's say, more stamens, then what does that impact? What does that mean in terms of, pollination strategies, right? Would they perhaps often use, let's say, insects for essentially promoting migration, or would they, and that regard, what types of other plants would they likely, fertilize? Could it be other eudicots, or could it be monocots in return? So those are things that you want to consider and the implications that it would have on their own reproductive success. Um, Now, apart from that, yes, so consider like, those different concepts, those details I touched upon before. Any questions, by the way? I'm a bit tight on time, I must say, but I hope, inshallah, that we'll be able to go through the material. Okay, perfect. Okay, inshallah. And, oh, get the screen paused. So let me fix some things. Okay, hopefully now you can see the screen. Um, so alhamdulillah, now we're actually, let me just unpause that. So, I'm gonna make an edit. I want to make sure that I finish on time. So, inshallah ta'ala, now we're at the stage of looking into plant morphology at greater depth and in terms of development as well from year one to week, right? So think about the things that I've talked about before for one to read, right? We talked about it briefly, but I try to provide a more Islamic approach from the scientific method because I have been able to study, alhamdulillah, for some time on campus, about 8 years, alhamdulillah. And so pretty much my advice is, taking a more authentic approach where you're not feeling demoralized, right? Because imagine you have a p-value that's 0.051, all of a sudden now it's rejected, right? Your alternative hypothesis. But in reality, things don't function by chance, right? I don't think like when I look around myself, likely when you look around yourselves in the zoo, in the lab, outside of the lab, around society, you can't just simply assume that everything around you is simply, less than a p-value or like essentially is governed by, you know, um, like pretty much random chance or like randomity essentially. So, or serendipity. And I know that sounds like a contrast with what's taught perhaps in lecture, but nonetheless, I personally believe that everything has a purpose, and this is something that indeed the Almighty has actually showcased to us. Where I was learning powerful dua and thinking about it earlier. Rabbana, so there's a group of people that, remind themselves or are remembrant of their Creator, their Lord, and they say in Surah Al Imran, أعوذ بالله من الشيطان الرجيم, الذين يذكرون الله قياما وقعودا وعلى جنوبهم. These are essentially a group of people that remember their Lord in the evenings and daytimes, and as they're lying down and like as they're standing in their qiyam, when they're sitting, and when they're lying down, like on the ground or on their beds. And they ponder وَيَتَذَكَّرُونَ فِي خَلْقِ السَّمَاوَاتِ وَالْأَرْضِ. And they ponder upon the creations of the heavens and the earth. And they say, رَبَّنَا مَا خَلَقْتَ هَذَا بَاطِلًا. Our Lord, you did not create this without purpose. Uh, فَقِنَا عَذَابَ النَّارِ. So protect us from the torment of the fire. And so I say this because, inshallah, these are things that, I want everyone to actually authentically understand and appreciate because, look around yourselves. Can it really come by chance? You know, Even the synchrony of your bodies, everything within yourselves. And that's also highlighted in the Quran, like to look within yourselves, to, seek the remembrance of Allah, the hearts find rest. These are things that, you look in embryology, zoology, astronomy, like pretty much every field imaginable, including economics and law and the governing society, or like thinking about society and the signs around us in nature, which is actually an amazing topic that connects us to AO2 and other bio courses. But you want to think about those concepts. And I know it may be difficult to understand now, but consider, having, consider thinking about more the creator of all creation, in terms of, um, eh, like, think about appreciate the creator over the creation because often if you think about the creation by chance and or like thinking about it in a nuanced way, you may lose your connection with the Almighty. And of course, you want to look at the creation, appreciate the signs that are given and governed by the creator, but don't lose that connection, is what I'm trying to highlight. So hopefully that's something that you can appreciate, all of you, and independent of where you are presently. So now that we're like considering this, in fact, right, when you're doing like experiments in the lab, you often want to control for certain conditions, right? And in reality, they're not always controlled for, but these are things that you want to highlight. So let's say, winter wheat, right? I know we didn't talk about it, but there could be other types of like serotypes or subtypes of plants, angiosperms of wheat that may have been, things that maybe you didn't account for. So let's say you're a farmer and you're trying to see, the effects of drought on your plants, on your winter wheat, for example. You may have a milia, like different types of winter wheat, like winter wheat A, winter wheat B, like different types of subspecies. And so those are things that you want to account for when you're doing like a controlled experiment, because one winter wheat subspecies may grow better and may be more drought resistant than another, right? Or some may even grow optimally at certain conditions. And that also is something that you want to consider. Sample size is another, right? If you're limited, like with a group of 10 different samples in one category, as we did from the pollen greens, then that limits the amount of not just variance, but of appreciation of, the diversity of different species or seeds that you'd be able to analyze. And so those are things that you want to consider. And then your independent variables, which is something I want you guys to be very cognizant about, because I notice that sometimes we limit our discussion regarding, the dependent variables, but think about them, consider them deeply, right? The differences in treatment for independent variables for that art, and then the dependent variables being your shoot biomass in units of grams and your shoot length in units of millimeters. Always remember to be very specific for your variables, for your controls, and for your species that you are analyzing. And the controls we've talked about before, including humidity, and actually they will be likely shown in an upcoming slide, but I just wanted to let you guys know that. And then when it comes to your research hypotheses, I noticed some of you may have maybe forgotten this, but the idea is briefly going through this. You want to have a specific um, and by the way, some of these have parallels, right? In the sense that, like, okay, I'm a Muslim, for example, and I want to be an amazing researcher. How do I balance my faith with, the profession in the sciences or with STEM? It's possible, and not even possible and doable. It is from the right approach, from the right angle and lens, perhaps even far more better than you would even imagine from the lens of the secular sciences. And I say this because as a student of knowledge, that you'll come to appreciate that taking like the divine approach will give you a divine not will give you essentially um, an edge up compared to your comrades. So like you'll pretty much be ahead of the game in every sense, inshallah ta'ala. So that's something to keep in mind. In terms of your statements now, um, you have like specific statement that refers to your species, the independent variables. If your hypothesis is testable, this is something that you want to also notice or take note of, and how it could be essentially proven, right? If it's proven to be correct or inaccurate accordingly. And so this is an example of what we had from week 3, right, with the Daphnia water fleas, right? So temperature would have an effect on heart rate. So temperature is independent, heart rate is your dependent variable. I actually recommend having both units provided, right? Because if you're in chem, there's kelvins, right? There's also degrees centigrade or Fahrenheit. So you may want to consider that. But briefly though, take note of the dependent variables with the units included, and then the species. And then normally, like in your FLRs, you provide further information as to how you're undertaking that hypothesis. And that's something that actually connects us to your predictions, which you likely also had in your intro. So your predictions are shown here as defined, measurable, connect to the hypothesis from the experimental treatment groups, and use a if then. So essentially, if your hypothesis is correct, then you predict XYZ, right? In comparing your 2, 3, or multiple conditions, right? So think about that. Think about like, let's say the winter wheat. If drought has an impact on the shoot or on plant growth of, winter wheat, and then in brackets the scientific name. Then as shown here within the blue category, or in the blue color, then I expect, or then may not put it in first person, but then shoot length and shoot biomass, both with units included, should decrease with increasing drought conditions or with increasing drought periods. So like comparing, the control with, treatments one or two, essentially. In the lab, FLR, you weren't asked to do this, but this is something you want to consider. The effects between of like analyzing the connections between drought treatment one and drought treatment two, right? Because you were only told to do stats comparing the control, right? And this is a question: was the effect of drought on the growth of winter wheat? You're only told to do this comparing to the control, right? But technically, in a more elaborate exercise, you would do it between all three, right? So the effective control, as done before, but in either treatment, but now you should also consider the effects between treatments, i.e., is there even a change in plant biomass between watering every other day versus every third day, or similarly for shoot length, right? And those are things with the variables below that you want to consider and controlling or accounting for. And so we talked about this already, but hopefully, if you have questions, then it hopefully is um, understood in this regard. And then essentially from your methods, which you likely also included, was about isolating plants in different categories. And this is something I want you to think about. Do you know why we actually chose six in the center, by the way, and not like seven or eight in the center and then six on either side? Anyone have a hypothesis as to why we did that? For Chidicum aestividum or aestivum? Okay, let's see. I will actually share for timing. But actually, if you want, you can also write it down, and then we'll compare our hypotheses. So, so I want you to think about your plants. So think about how they were set up. Imagine just above you had the light source, right? Which should be controlled for. You want to think isn't as well as the mid side as the sides. So if you think about it, the middle will likely get direct exposure, right? More exposure generally if the light source was in uh like essentially the middle. So generally speaking, you may have an overestimation then, and even accounting for seeds that maybe were distributed from the center but not fully evenly would technically have a normal distribution where essentially plant biomass and plant shoot length would appear larger in the center because there's more nutrients, there's more seeds perhaps, there's more light exposure primarily. And then at the edges is where you'd have less of that, perhaps, right? Or even just the amount of water that is sucked up, which is differential from the edges because you don't have the other sides, like you don't have water from another side at those edges to account for. So essentially, it's likely limited at the edges. So to average and to get a more representative um, random selection, you would actually get a little bit more on either side. And so that's something that you want to consider. And then in contrast to the effects of having too many seeds in the center, which is a limitation that you may have touched upon if your group had that concern. But briefly, actually, I want to go forward, in fact, to save time, inshallah, in that you would have essentially done the shoot length and shoot biomass analyses, right? We saw them on Quercus, and hopefully you were able to essentially account for the mean for all 40. So it wasn't just 20 seedlings, by the way. This is like a mini version, but technically you would have done this for both reps, right? Because each half of the room of our lab had essentially all three treatments. So they were replicated twice on either side. And so you would have had 40 seedlings to account for and the shoot biomass versus shoot length, and consider the differences between the two, right? Because um, actually, I'll go to exercise two with that in mind. Inshallah, if there's questions, let me know. But as that concludes the FLR, we're now looking deeper into the tissue layer of plant organs. And we looked at celery stalks, but knowing their functions as just plants in general is something that connects us even back to the FLR, right? They need light, they need oxygen, different nutrients and byproducts shown here. And, these are things that you want to consider in mind, including water. And also keeping in mind the photosynthetic biochemical equation, right? So you have essentially um, CO2 plus nutrients like carbohydrates, sorry, CO2 plus water, uh, thereby leading to with light as a catalyst or as a source from Allah subhanahu wa ta'ala, leading to oxygen by His mercy and water to be produced. Well, oxygen and carbohydrates. And these different equations, by the way, I know they sound like a given, but in reality, there are different ratios that come into account. And really, when we're looking at the different organs, we're looking at the different methods or different steps in which photosynthesis and different cellular or biochemical reactions occur within the cellular level. And so we have the shoot that you would have cut out, by the way, for the biomass analysis for the FLR for when to reach. So that's a nice connection here. And then, the effect of roots is something I noticed some of you also touched upon in your FLR, which is also a good point to consider. Stem, this is maybe something to take into account, right? For the celery stalk, as we did a cross section, there are different importance points to take into account. One is stability, right? It keeps your plants upright, you know, as like there was a word, an Arabic word I'll touch upon, Hanifa, like upright and firm. And so essentially you have a stem that is, founded with being able to stand firm, even, with taking into account that there may be other organs that are above, right? So it's balancing with turgid pressure, like there's different components at play. One is, stability, another is nutrient flow and byproduct flow, right? Because of the different biochemical photosynthetic reactions that are at play. And then also, maintaining turgid pressure or like maintaining pressure and balancing or like homeostasis, but at a more pressure turbid level, or a more like physical level to essentially maintain its balance in the outside of the ground, like overground, and also being able to essentially interact, in fact, with other organisms like ants and other organisms that may also try to go up to pollinate, right? As we talked about insect pollination. Um, Now, when it comes to examining these certain plants, you want to also keep in mind the plant stem, right? So you would cut a thin layer. We use the microtome for this and then stain with toluidine blue O or TBO stain. And so you would have used this, and you would have seen differences in colors, hopefully, right? Comparing the pinkish purple color. Actually, let me just see if there's more. Okay, this is good. We're at a good pace, inshallah ta'ala. Sorry, the TBO stain, as you can see, blue highlights, one level of coloration, and that's usually used for the secondary plant walls because it includes cellulose plus lignin. Lignin gives it a bluish green hue, and then the primary cell walls, which include more of the other types of components, usually stain in pinkish purple, right? So you want to consider those different components and what you've seen from the lab manual or even from online to see, okay, from the six different components, which one of, like knowing their functions, yes, but also knowing as well as to what major component they play or serve in the celery stalk or in your other plants of interest, right? So are they primary or are they secondary? We talked about for your presentations, what size they have, what functions they play, and the different shapes or arrangements that they also have. So those are things that will help even connecting it to histology, because histology is a subfield or a specialty where you look into the differences in tissue morphology, which has pathobiology relevance, but also like medicinal, right? If you see like a tumor or a tissue where you have differences in cell shape or size, maybe that could give you an indication of to what kind of condition your patient has, depending on where you found or where that sample was taken from in a given patient. So those are things that you want to consider as well, connecting the dots once again. And then pretty much connecting back with the cell size. Now, in this regard, not really with the pollen grain, but you would have looked at 40x objective lens. So your calibration factor would have been multiplied by 6. So let's say, I'm just going to give an example. Let's say your epidermis layer was 10 microns. Sorry, um, well, I said, let's say it was 3 OMU. What would be the number of microns that I would have at 40x? Remember, you need to know the magnification or the objective lens. But what would you assume then for calibrating the size of your epidermis? If it was 3 OMUs, 3 ocular units long. I'll let you know. So if it's 3 OMUs, you multiply. So this is just an example, right? Or for helping to actually calibrate the okay, no problem, no problem. I'll try to go over it to like share where I'm coming from, so that way the methods are clear, inshallah. So so these are different components that you would include for your table to actually derive the amount of to determine the calibration factor, which is thereafter converted from millimeters per OMU to micrometers per OMU. And so if you have three OMU, right? So think about the calculation. If you have three OMU times 6 micrometers divided by OMU at the specific magnification of 40x, then that would be 3 times 6 gives you 18. And then when you think about the units, OMU time divided by OMU is going to be 1. And then what's remaining at the numerator is microns or micrometers. So you would have in total 18 micrometers, which would be the thickness of your epidermis. Generally, you would have had them around like 1 to 2 OMUs, which would reflect a more conservative um, a size of about, sorry, let me go down. uh, a conservative size around 10 micrometers. But nonetheless, I hope inshallah ta'ala like these different features you were able to take into account and hopefully understand, especially when it comes to your lab exam, because you may want to consider, differentiating certain components, for example, the parenchyma from the sclerenchyma, the parenchyma from the chlorenchyma, or because the sclerenchyma we didn't have from the celery stalk cross sections, but then you would have primary versus secondary structures and comparing like the different components and what they serve as shown here. Um, so that's something that you would have done. That actually concludes, inshallah ta'ala, lab 8, sorry, lab 3, lab 2. Um, any questions? Let me actually return back to the previous or the upcoming, inshallah, the upcoming slide. And this is with regards to, let's see, lab 3. Probably one of my favorite. And in fact, I actually did this in undergrad. So it was so amazing being able to actually literally see the heart beating, like see through from a kilotherm, as we are aware. And alhamdulillah, it's a profound creation of Allah that, subhanAllah, you're able to see like these organisms beat, change in heartbeat and like, in different levels, right? Or at least across different temperatures. And so another thing, like bearing in mind, oh, the Toronto Zoo. I hope you guys enjoyed it, by the way. It was really nice. I really hope you enjoyed it. But when we look into metabolic rate, and this probably connects with Stephen Reed's lecture material because it reflects physiology, animal physiology, and metabolic rate or animal metabolism. And so, as we, contrast to what we've seen in plants, right? They have specific nutrients that they need. Here, we're looking at the output, generally from what organisms are able to essentially biochemically reduce or make use of in the form of energy. And so now that, let's say, oh, my timer just went off. Sorry. As we continue, inshallah, I want you to think about, the energy demands of organisms, the changes in external or internal temperature, right? This actually connects us. Like I really hope you consider the demands in connecting, these different concepts because, you know, um, like different organisms may reflect different demands, like birds versus turtles, right? Like eagles, like the American bald eagle, or like like changes in temperature, which as we saw has different effects in different organisms based on their own patterns. Or actually, as we go here into Daphnia subspecies, we know they're crustaceans. Probably where we'd find them is in Australasia and they're water fleas. They're poikilotherms. And I want to highlight this because their internal temperature mimics their external. And so, as we see, hopefully, from ectotherms, they are also influenced by the environmental temperature. But what makes it unique for poikilotherms is that the rate of change is more drastic, right? Or it's more sensitive to changes in external temperature. So you could see, like, as you change the temperature in 10 or 5 centimeters, 5-degree increments in centigrade, you should hopefully see the change substantially in heart rate as you could probably see even from this annotation here. And so that reflects also changes in not just heart rate, but perhaps other components. Sorry. Essentially, you want to keep, let me see actually. Yeah. So just sorry, keep those different details in mind, right? So like the changes in temperature. And something to bear in mind that the heart is actually down at this region, right behind the gut, the gut lining here. And so let me just actually go down. I just want to see. Okay. Um, sorry for the blip. Let me just see from this. I don't know if we have a video. Okay, this is water flea heart beating. So I think we actually showed this, and it's so beautiful of it with accounting for these vertebrae at different temperatures. You want to be able to record with a counter the heart rate, right? So how many beats you found every 10 seconds? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33. Let's say it's 33, right? Then you would multiply that by 6 to give it in a minute for every 10 seconds. You had 33. So that'll give you probably 198. That's actually very quick. But as you can imagine, right? Like I'm saying quick in the sense that like imagine, subhanAllah, like having 3 beats every second. That's profound, but that's what we're seeing here, right? Um, in that regard, you want to compare, right, like with the literature, and even comparing with ourselves, right? 70 to 80 beats roughly per minute is what we see. And then comparing that with, what you actually saw is something of importance. But essentially, sorry, I don't want to tangent back, but you would start with room temperature. Everyone knows that, like the function of beginning with room temperature is also to control, right? To have a baseline for your specific Daphnia because they are different to a degree, right? And so the idea is that you want to count essentially every 10 seconds. I said 10 seconds and then multiply by 6. It's similar in this regard. But the way I did it, it gives you the heart rate per minute, which is also more appropriate, which is more perhaps relevant compared, or perhaps um, is also beneficial. Sorry, it's also useful to take into account. But you essentially start at room temperature, right? So 25 degrees Celsius. And then you go down, right? Using the ice cold water, slowly going down, seeing with the thermostat, going to 10 degrees, then adding in hot water that was left at the water bath in the middle bench at 55 degrees, slowly adding in from the small beaker into the large beaker to make sure you get the right amounts of temperature that you have added in using the pipette, taking out any extra remaining water. Generally, you wouldn't add too much, but taking into account that you have a small region where your single Daphnia is located, where the Vaseline is also present. So you just want to add in slow droplets amount at a time, maybe 3 at max, 3 to 5 at max, within that range or that within that region. And so you would have done that, seen the changes at 5 degree increments, and then you would have actually recorded this data to what you would have seen across the heart rates, right? And then you would have averaged them to collect the mean, and then you would have gotten your data analysis, right? And so that's where you would have actually calculated the heart rate, and then you would have actually compared based on what you see actually in the lab manual. I think that would make it easier. Sadly, I don't see it here, but okay. So pretty much you would have two sets of formulas. Make note of both because you want to know if the difference between the two temperatures that you would have compared are 10 degrees apart or less. If they're 10 degrees apart, if they're less or more than 10 degrees apart, calculating the Q10, which is your stat for the relative impact or change in rate of a biological, so like the impact or the sensitivity that a biological reaction has in relation to temperature. That could be heart rate, that could be fur growth, or in Arctic wolves, for example, etc. And so, but the idea is that you are comparing across temperatures. And so there needs to be, if there's less than or more than 10 degrees of a difference apart from both rates that you're analyzing or the different measurements that you're analyzing, then you would use this log based formula shown in your manual. And then if it's exactly 10 degrees, let's say 20 to 30 degrees on the dot, then you would generally use this ratio of the higher, like generally speaking, the reaction at a higher temperature or at a certain temperature compared to divided by the rate of the reaction at a 10 degrees lower temperature. So it could be like fur growth, right? Or it could be even like, let's say, shoot biomass, right? Like, think about what we did for FLR, or even like shoot length, right? So shoot length is also a metabolic reaction. It's a biosynthetic, photosynthesis is something we're referring to, but biological reaction is included in this regard, right? So even for plants, this is similar in the sense that let's say you were looking at differences in temperature of the water that you added and continue to add, let's say, every other day, continuously for your winter wheat, then you would see, let's say, comparing the results at 10 degrees versus 20 degrees, water being added in, and seeing the results, maybe 2 days or maybe 2 weeks later, you would hopefully see the differences in, shoot length and then record it from the tip of the longest shoot, as we talked about, to the very bottom before you cut the seedling, or just at the end before you touch the seed. And so that's where you would essentially record, let's say at room like the shoot length at 20 degrees divided by the shoot length at 10 degrees. That will give you a Q10 value. And then your Q10 relates to essentially the sensitivity. If it's closer to 2, that means it's more sensitive, like in the case of the poikiloderm, Daphnia, right? Or if it isn't, let's say closer to 1, then it indicates that it's relatively less sensitive. So you want to see that there's a spectrum between 1 to 2, generally speaking, and the higher the value, the more sensitive it is based on that specific condition. Um, I'm gonna end off there actually, inshallah ta'ala. Are there any questions? No? Okay. No problem. Yes, I plan to do so. I just also want to ask I want to spend a bit of time if there's any questions. No questions? Okay. Okay. So let me go over maybe okay, give me one moment actually. I need to breathe a bit, sorry. Um This is actually something to keep in mind. I didn't know how valuable this uh, app is, by the way, if you guys were using it, for even if you have future like labs to tally count, it's actually really interesting. So you go online, and then it will essentially give you a timer, counting like every 10, 15, or 30 seconds, and you just keep counting with your head, and it will tell you, oh, it's 10 seconds. So you just record that value rather than having to depend on both a timer and a counter. And I was actually amazed. So some groups, I know a group that was using this during the lab, which was found very interesting and quite beneficial. But nonetheless, um, yeah. Let me go over like going back to the different topics and maybe um, no, I don't know if I can do this, but let me just go over maybe some concepts that we talked about to give you like an understanding as to what you want to consider, right? You want to consider the intro, right? Like an FLR. For this assignment, in a sense, this assessment for the final exam or your lab exam, you want to consider the intro, introductory concepts, material, the key terms that we discussed, what you see in the lab manual, maybe connecting back to your lecture slides, but generally focusing on what you have available. You want to know the material, right? You want to know it pretty well. So when we were talking about lab one, what is the winter wheat? Being able to know as well, like its scientific name, the different components we talked about as well. Um, Let me just go, for example, the flower morphology, right? You want to know all six different components. I believe maybe even seven, including the eggs themselves, right? But you want to know the different components that we talked about, and being able to do stats. Actually, I'm going to go back so you can understand where I'm going with this. You have the intro, right? For the different labs that I've talked about thus far, from morphology to ultrastructure, like cell cross sectioning, and then the physiology of Daphnia, right? And then you have those intros, and then for each of them, you have methods, right? From those methods, you want to explore, okay, what am I measuring? What am I controlling for? And what variables am I, or sorry, from the hypotheses that I made or that I'm making from the intro, um, what steps or parameters am I thereby measuring or seeing, like analyzing with specific predictions in mind, right? So as you do these exercises, even for example here, you should be able to consider, okay, I know Daphnia from the intros of kilotherm. So if I'm adjusting its heart rate, what should I see as a research prediction? Your hypothesis would, revolve around if, as we saw earlier, temperature would have an effect. Temperature in degrees Celsius would have an effect on heart rate, measured in beats per 10 seconds. Um, in Daphnia, SPP, water flea, right? Or like in the water flea, common water flea, brackets, Daphnia in italics with SPP dot. So you would have that. And then you have your predictions, right? If it is expected that, my hypothesis is correct as an alternative, right? Your null would assume that there isn't. But if your prediction, within your prediction, or if your hypothesis is correct, then you would predict XYZ, right? So you want to consider that, okay, what effect then would temperature have, across the different conditions? You probably would see at 10 degrees, the lowest, heart rate per 10 seconds, or heartbeat rate per 10 seconds, and then the highest maybe at 35, or the highest temperature that you're looking at. And that also reflects, the different optimal conditions, right? Because if you look at other species of organisms, and this maybe connects us with Lab 4, inshallah, for Thursday, بإذن ta'ala. Thermoregulation is also something you want to keep in mind. Some organisms are able to grow, survive, and live at higher temperatures. Let's say in like the Sahara Desert, like the bearded dragons, or like cacti, for example, as well, versus versus at colder climates like the Arctic wolves, right? Or the African penguins, or the snow geese, right? So these are things that you want to consider, and thereby understand the relevance and the pursuit of results accordingly, like how you would essentially measure certain conditions, how you would record and control for certain variables that may be unaccounted for, right? Like the species, as we talked about, the sample size, the other external factors like temperature, humidity, etc. So, and light exposure for plants, right? So these are things that you want to consider, and those relate to your methods. And then the results is what we've talked about quite a bit in terms of, you know, sorry, pretty much we've actually already spoken about it, so maybe that would suffice. But uh, you want to be able to, like observe and then record the data that you've observed, right, from the different measurements that we've described thus far. Um, so those are things that you want to take into account. Um, Lab exam, sorry, I just saw your question, Alameen. The lab exam is going to be diverse in a sense. So you have, if you look at the last lab slides, you'll have multiple choice, multiple, like select all that apply, numerical answers, matching questions. So these are the ones that generally speaking you'll have. So they may be a bit, they'll vary in, I would assume they vary in size. So you just want to bear that in mind. Um, in terms of understanding, and that's why it's important. You want to, like, you want to succeed in understanding the different components I talked about thus far, from the intro to the methods, like the hypotheses that we talked about, the key terms, hypotheses, predictions, into your methods, the measurements, the controls, the observations that are then needed toward, or sorry, into your results, where you actually talk about your observations in greater depth and the results of your measurements, or from your figures or tables or graphs, etc., with the detailed figure captions, as I mentioned before. Then you have the stats, right? And actually, when you're done, essentially, you'd have your discussion, right? And that's very important as well, because you're now interpreting the data, right? From observing them in your results, or actually from introducing the topic of your interest, right? The experiment as a whole, to the methods of undertaking that exercise, to then the results of observing those data, like your data or your findings, or like your data, to then into your discussion, interpreting that data, analyzing, extending, or analyzing you're doing your results, but extending the implications and limitations or connections of your data to in your interpretations, interpreting them in line with or connecting back to literature, to STEM and beyond. These are things that you want to consider, and even any limitations that you would like to further, either correct or see, like analyze further in depth to see if you can, extract further analysis and appreciation of your um, introduced topic of interest. So you want to like essentially understand the different concepts. And let me just, within the last technically 19 minutes, I know I said earlier about to 12. We actually finished, alhamdulillah, a minute just after, but I want to make sure that if you guys are actually in need of me going back, how about that? Let me know. Pretty much in terms of stats, we talked about this, but you want to understand that 95% confidence intervals reflect your mean in which you are 95% confident, like the semi-confident at least, that your mean represents the actual average of your data points or reflective of your category of interest. So, or of your condition. So this not only is like the ones shown here, but of other categories that you've done thus far. Um, if there are any other questions, I'll end off shortly, inshallah, but I'm just going to see if there's any outstanding components I may have missed. Don't forget for calibrations, look at the table, focus on the magnification, going down to the calibration factor, noting the units. If it's shown in millimeters, you may have to multiply by 1,000 to get in microns. So just bear in mind that you follow, based on the lab exam, specific instructions that may be provided. And also bear in mind that it's, if I'm not mistaken, it's closed book or individual, so bear that also in mind. Um, but you could see the details on Quercus as well. And it's only 1 hour, just to clarify. But then briefly, this is where I didn't talk about it, but okay, let me see. Yeah, so like you want to also understand and appreciate the settings of your organisms, be it from, the lab in of itself of the winter wheat, the celery stalk, the Daphnia. These are all different types of organisms, believe it or not, right? So they have different traits in morphology and thermoregulation and, in their own physiologies and their cellular structures. So these are things that you want to understand and account for when you are examining and comparing different results across conditions, right? So you want to consider the implications as such, as I've described earlier, and also be mindful of the conditions that are being mentioned. Let me just think about the celery stalk. So, you wouldn't over dye, right? With the TBO stain, right? You'd only add in maybe 1 or 2 drops on the surface of your stalk, and you would have had, or on your, yeah, on your cross section, and it would be generally quite thin, right? From the microtome. So bear that in mind. You wouldn't want to have it like staggered, i.e., like at an angle when you're cutting, but so that way you can actually represent, like accurately see on a focal plane your cross section of interest. Um, Maybe for the Daphnia as well, making sure that you've added in the right amount of water, not overdoing it, not over adding Vaseline, and even another point, bearing in mind that you're seeing the right organ, right? And like with the contrast that was done with the black layer that you saw also from the video, you can kind of appreciate that, seeing it at greater contrast provides a better angle to interpret the heart rate, right? So it was under a black disc with the contrast of the light above. So you would be able to more accurately record the heart rate. And then there was also sensitivity to light, right? So those are things, those conditions connect theory to reality. And I want you to extend those concepts and what I've talked about from the I5 approach of Islam, integration, investigation of inventions to implications, to actually see what you're able to surround yourself, understand the concepts of, the lab material, to extending them to other questions, other concepts that you may see and come to know soon, inside and outside of your lab exam. Try to understand these concepts, and I really hope you'll have a due appreciation or come to, you know, um, understand readily the different concepts and integrate them accordingly within your life and beyond. So, with that, I appreciate your time. Any final questions? I hope you do too. And no problem وإياكم. إن شاء الله تعالى I'll conclude. سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. Inshallah ta'ala, looking forward to seeing you for the review of labs 4 to 6, inshallah, on Thursday, 3 to 5 p.m. EST. Same link, inshallah ta'ala. Take care, all of you. Really appreciate the time. Assalamu alaikum wa rahmatullahi wa barakatuh.